Embryonic circulation. This lecture concentrates on the processes involved in establishing embryonic circulation. After the third week of development, the size of the embryo is such that it can no longer rely on simple diffusion to distribute oxygen and nutrients to all of its cells. It must therefore quickly develop an efficient means for the distribution of nutrients. This occurs via the development of three main vascular channels within different areas of mesoderm. Those that develop within the extraembryonic mesoderm will be discussed within this slide. As a review, the image to the left highlights appearance of the extraembryonic mesoderm during the second week of development. Following initial formation of the yolk sac, the hypodermal cells then gave rise to the extraembryonic mesoderm cells. The mesodermal cells multiply and spread to surround both the amniotic cavity and yolk sac. Development of the blood vessels first occurs within the extraembryonic mesoderm that covers the yolk sac. These arteries and veins are referred to as the vitelline vascular plexus. Shortly after formation of the vitelline vessels, vascular development progresses to the extraembryonic mesoderm of the connecting stalk. These primitive vascular channels soon form the right and left umbilical arteries and umbilical vein. Within the embryo proper, a series of arteries and veins also develop within the non-axial populations of intraembryonic mesodermal cells. As a reminder, non-axial mesoderm reflects those cells that have differentiated into either somites, intermediate plate mesoderm, or lateral plate mesoderm. Note also in the image the presence of the dorsal aortae that were formed via the non-axial mesodermal tissue. As an aside, the aorta first develops as a bilateral structure, but we will discuss aortic development further in the next slide. By day 22 of development, which is when the embryo is just entering into the fourth week, blood is being pumped through the vessels by a primitive heart tube, which formed as part of the embryonic folding events. There are three main routes in which blood is returned to the developing heart. At its caudal end, the primitive heart tube receives oxygenated blood from the developing placenta through the umbilical vein. It receives deoxygenated blood via two sources. Blood from the yolk sac is delivered via the vitelline vein, and blood from the body of the embryo through cardinal veins. Notice that all three of these venous channels are colored in blue, signifying the fact that they are veins, not providing indication to their oxygen content. Out of the three, the umbilical vein is the only vein carrying oxygenated blood. Now we will consider the route in which blood leaves the developing heart. Upon exiting the heart tube, blood passes dorsally through the pharyngeal arches via aortic arch arteries. The blood then reaches paired dorsal aortae that pass along the entire longitudinal axis of the embryo. Near their caudal ends, the dorsal aorta give off the umbilical arteries, which carry deoxygenated blood back to the developing placenta. Again, note that they are colored red because they are arteries, not due to the content of oxygen that they contain. With subsequent development, the two dorsal aorta will eventually fuse in the abdomen and in the thorax regions to form the definitive aorta. The two umbilical arteries will persist as paired structures. Blood formation begins in the extraembryonic mesoderm that lines the yolk sac. Remember, this is the same mesoderm from which the vitelline vascular plexus is derived. So blood islands will begin to develop within this layer, and these serve as the initial factories for making blood. This continues for most of the embryologic period. By week 9, however, the liver and spleen take over blood production. This concludes the lecture on embryonic circulation.